Hello, today I'm going to go over active transport, endocytosis, and exocytosis lecture. Now, we've been talking in the past about moving things in and out of the cell, but moving them in and out that doesn't require energy. It's passive. This is going to be the opposite. All of these processes are going to require some sort of energy for them to happen. So let's get started. Active transport is the movement of substances across a cell membrane against their concentration gradient. This is the big one, against their concentration gradient. So substances can move from high concentration to low concentration without any energy input. But to get them to go the other way from low to high requires an energy input. Because you're putting things closer together. They're energetically less likely, and it's not as favorable for them all to be coming back together. So you have to put energy in to make it happen. This process requires energy, usually in the form of ATP, because molecules are being moved from areas of low concentration to high concentration. Specialized proteins called pumps or carriers are embedded in the membrane to help move these substances. You can see this here. So here's our plasma membrane, right? The skin of the cell. And then what it's showing here are proteins that are going to act as pumps or carriers, moving things against their concentration gradient. And that's what you see here. The sodium comes into this one. You get a conformation change because of an energy input, and then they're expelled out into an area of high concentration. A well-known example is the sodium-potassium pump, which maintains proper ion balance in cells. That's what we have here. Sodium, potassium. Okay, let's continue. Active transport is essential for maintaining homeostasis within cells and across tissues. So balance, right? Things in the right order. It allows cells to take in necessary nutrients even when they are in lower concentration outside the cell. It also helps remove waste products that might otherwise accumulate. Without active transport, many cell processes would fail because concentration gradients would not be properly maintained. So again, we have this simple diagram here showing the cell membrane and a protein spanning it. This protein is going to move whatever this molecule is in red against its concentration gradient, where it's higher outside than it is. Where it's higher inside than it is outside. So it's going to move them in using ATP. And again, the big thing is against the concentration gradient. Endocytosis is the process by which a cell takes in large particles or molecules by engulfing them with its membrane. And that's what you're going to see here. There's some stuff that's going to go into the cell. Endocytosis is going to use the cell membrane to wrap itself around those molecules. The membrane folds inward to form a vesicle that encloses the material being brought into the cell. There are different types of endocytosis, including phagocytosis, cell eating, and pinocytosis, cell drinking. This process is important for nutrient intake, immune responses, and cell signaling. So again, another type of active transport. And then in this one here, we have more on endocytosis. So still taking molecules into the cell, but wrapping them in cell membrane. Endocytosis allows cells to interact dynamically with their environment. For example, immune cells use phagocytosis to engulf and destroy pathogens like bacteria. Receptor-mediated endocytosis enables cells to selectively absorb specific molecules such as hormones or chemicals. So one way that they do this is there's a receptor on the surface of the cell, and when you get that thing you want in here, when this ligand lands on that receptor, then it's going to start this process of endocytosis surrounding it. The vesicles formed during endocytosis can fuse with other organelles like lysosomes for digestion. Well, here's a lysosome, and it's used to break down things in the cell. So you're recycling, right? Well, this vesicle is going to merge eventually with a lysosome here and here to break down whatever material this stuff was. That's just one example of what could be going on. This makes endocytosis a highly controlled and efficient process. All right, let's continue. The opposite of endocytosis is going to be exocytosis. Exocytosis is the process by which cells release substances to the outside environment. So now we're going from the inside out. Vesicles containing materials fuse with the cell membrane, allowing their contents to be expelled. So you see this vesicle in here with some kind of, in this case, waste, which would make sense. It fuses with the cell membrane because it's made of the same stuff. And then the waste in this particular case can just be released out into the extracellular fluid, the outside of the cell. This process is essential for removing waste and for secreting important substances like hormones and neurotransmitters. Exocytosis requires energy, like all the other ones, and is carefully regulated by the cell. All right, so the last little bit here on exocytosis. Exocytosis plays a major role in communication between cells, especially in the nervous and endocrine system. So this here is the head of a neuron. So the neuron would go up this direction, and this is just kind of the head of it, where there's going to be another neuron right here, and messages will pass back and forth between these two using chemical signals. Okay, so that we'll learn more about this in a different section. But when we talk about exocytosis, this is a prime way that these two neurons, like neuron 1 and neuron 2, are going to communicate back and forth is by excreting or exocytosis these chemicals and neurotransmitters. Okay. For example, neurons release neurotransmitters into synapses through exocytosis to transmit signals. Similarly, endocrine cells secrete hormones into the bloodstream using this process. The cell membrane expands temporarily as vesicles fuse with it, but it is later balanced by processes like endocytosis. So even though during both of these, exo and endo, you're going to have, in some cases, you're going to be adding to the cell membrane. In some cases, you're going to be subtracting from the cell membrane. But the cell membrane can maintain itself. That's not a problem. It's not going to vastly shrink or expand in size because of these vesicles either going in and out. Okay, I hope this helps. This gets us started. Let me know if you have any other questions. Thanks.